Today we're going to be looking at exceptions to the expected Mendelian phenotypic ratios. And so it's important to understand that the actual inheritance here doesn't change. You know, we have two sets of chromosomes, we pass on one. That idea doesn't change, but what we're looking at here is where we don't see the phenotypes we expect to get under certain conditions. And so, you know, why are those things occurring? So one of the things that can affect expected phenotypic outcomes is something called epistasis. Epistasis occurs when one gene interferes with or actually controls expression of another gene. So we actually are looking at interaction between two different genes here. And so when we have an epistatic situation, there's an epistatic gene, which is the controlling gene, and a so-called hypostatic gene, which is the one being controlled by the epistatic gene. So one of the situations where we see this in nature is actually with horse color. And when we look at horse coloration, there are a lot of complexities to horse coloration, and there's kind of a basic starting point here, and that's where we're going to begin. And so when we look at basic horse colors, there is essentially black pigment and brown pigment. And yes, I understand there are a lot of other variations of that, but those are kind of really the basic starting points when we look at horse coloration. So one of the genes involved in horse coloration, as we look at this brown and black pigmentation, is what's known as the agouti gene. And in the epistasis relationship, the agouti gene would be what we call the hypostatic gene. This is the one that's being controlled. So we really have two possibilities here, or two alleles here, as we would have with any gene. And in the agouti gene, the dominant allele codes for black pigment, but black pigment that is restricted to what we call the points of the horse. And the points of the horse would be the mane, the tail, and the legs. And so a brown horse then that has black at the mane, tail, and lower legs we refer to as a bay horse. So when we look at the agouti gene, if a horse has the dominant allele, what it's saying is make black pigment, but make it a bay horse. And then a recessive allele here is telling the horse to make black pigment, but make it everywhere. Don't restrict it to the points. And so that would be coding for a black horse. So again, dominant allele, bay horse, black pigment at the points. The recessive allele produce black pigment everywhere, which would make a black horse. Now, expression of that gene, whether that gene is going to function or not, is controlled by another gene, but I'll come back to that in a second. So that when we look at the agouti genotypes then, if a horse is homozygous dominant then, it would be saying, or heterozygous, as long as there's a dominant allele present, it's saying, I want you to make a bay horse. And then if the horse is homozygous recessive for the agouti gene, it's saying make a black horse. But whether black or bay can be coded depends on the other gene, the epistatic gene. And in this case, that epistatic gene is called the extension gene. So the extension gene also exhibits complete dominance and has two alleles. And so here the dominant allele is telling the agouti gene that yes, you can function. So yes, you can make black pigment. And it would depend on whether the agouti gene, or whether the horse was homozygous dominant or heterozygous as to whether it would make a bay or black horse. But the extension gene is saying, yes, you can make black pigment and you can make either a black horse or a bay horse. Here, the recessive allele is saying, no, I don't want the agouti gene to function. Do not make any black pigment. And if no black pigment is being made, then what we're left with is a brown horse, a chestnut horse. So again, as we look at the genotypes here then, if the horse is homozygous dominant or heterozygous then, it's saying, no, I don't want you, or excuse me, it's saying, yes, you can function agouti gene. The agouti gene can function, so go ahead and make either a bay or black horse. But if the horse is homozygous recessive, it's saying, no, I do not want the agouti gene to function, no black pigment being produced, therefore it's going to be a chestnut colored horse. So as we look at the combinations of these different genes then, and genotypes, and the phenotypes that they produce, a bay horse then, as long as there's a dominant agouti gene or allele rather, and a dominant extension allele, then you'll have a bay horse. So for example, in this situation, homozygous for agouti, so it's saying I want to make a bay horse, and so the extension gene is saying, yeah, go ahead and make a bay horse. Same thing here. I want to make a bay horse, go ahead and make a bay horse. I want to make a bay horse, go ahead and make a bay horse. So again, it's the extension gene that ultimately makes the decision here, but these combination of genotypes then would generate a bay horse. A black horse would occur as long as you have homozygous recessive for the agouti gene and then a dominant allele for the extension gene. So again, this is saying, I want to make a black horse, and the extension gene is saying, fine, go ahead, make a black horse. Same thing, I want to make a black horse, go ahead, make a black horse. And then finally, chestnut horse. This would occur anytime that the extension gene is homozygous recessive, because it's going to be inhibiting the agouti gene. So here the agouti gene is saying, I want to make a bay horse, but the extension gene is saying, no, you cannot function, you cannot make any black pigment, so we end up with a chestnut horse. Same thing here. Or excuse me, bay horse actually. I want to make a bay horse, and it's saying, no, you can't make any black pigment. So again, I want to make a bay horse. Nope, sorry, you can't make any black pigment, so we end up with a chestnut horse. Here I want to make a black horse. Sorry, no black pigment allowed. And so let me go back because I didn't mean to jump that quickly. So here again, we're looking at the genotypes then that would code for a chestnut horse. So let's go ahead and take an example. Let's go ahead and look at a cross. Let's say we have a bay horse. So it's saying I want to be bay. Extension is saying fine. So homozygous dominant with heterozygous here. And let's say we've got another bay horse. I want to be a bay horse, and it's saying yes, be a bay horse, but heterozygous for the two genes here. So what we would do is approach this problem just like we would any other cross where we're looking at two different genes. So we want to look at independent assortment, assume independent assortment. So if we look at homozygous A times heterozygous A, we would expect half of the offspring to be homozygous dominant, half to be heterozygous. So we would put our outcomes on one axis. And then we would look at the extension cross, heterozygous times heterozygous. And if we cross heterozygous, we get a quarter homozygous dominant, half heterozygous, a quarter homozygous recessive. And so again, just like any other independent assortment problem, we would multiply to see what our outcomes would be. And so we would expect an eighth to have this combination of genotypes, an eighth to have this combination of genotypes, a quarter, and an eighth, and an eighth. And so if we looked at this then in terms of phenotypes, we would add these together, particular phenotypes together. So if we're looking at bay horses, we would have an eighth and an eighth, which is a quarter, plus 2 quarters, plus 3 quarters. We would expect 3 quarters bay in this cross, and 2 eighths is equal to 1 quarter. We would expect 1 quarter of the horses to be chestnuts. So again, you could look at particular crosses of different types of horses and then predict phenotypes. But the point here is that even if we were crossing dihybrids, horses that were heterozygous for both traits, we would not get a 9:3 to 3:1 ratio here because there are only 3 possible phenotypes based on the interaction of the 2 types of genes. So this is what I mean when we see exceptions to the expected Mendelian inheritance in terms of phenotypes. The actual inheritance isn't any different. It's just that because of the interaction of the genes here, we don't end up with 4 distinct phenotypes. We only have 3 possible phenotypes. All right, now another situation where we don't see expected phenotypes in all situations involves inheritance in animals. And one of the first researchers to investigate this was T.H. Morgan. And I've mentioned before, I think, that Mendel was not really understood or appreciated in his own time in the mid to late 1800s. And his work was forgotten. And anyway, T.H. Morgan began his work in the early 1900s, and he and his compatriots rediscovered Mendel's work, understood its significance and so on, but wanted to know if the same kind of inheritance occurred in animals. And so T.H. Morgan was working with animals. And he was actually working with something called the Drosophila fruit fly, and that became a staple of genetic studies for many, many years. And other types of organisms are used now. We still use fruit flies, Drosophila. We also use things like nematodes. Caenorhabditis is a type of nematode worm. Zebrafish are also used quite a bit. And what these all have in common is that they're small, they reproduce rapidly, they're easy to keep, and again, are really convenient to do genetic studies with. So anyway, as Morgan was looking at inheritance in animals, most of the time he saw the frequencies that he expected to see, but occasionally he did not see expected phenotypic frequencies that he expected to see, and that these had to do with sex more often than not, that males and females did not always show exactly the same types of phenotypic ratios. And so what came out of those studies eventually was the realization that in animals at least, that there are specific types of chromosomes that are sex chromosomes, chromosomes that determine the sex of a particular animal. So when we look at our chromosomal makeup then, there are autosomes. Autosomes are the non-sex chromosomes. And so in the case of humans, we have 23 pairs of chromosomes, as we've talked about many times. So chromosomes 1 through 22, or chromosome pairs 1 through 22, would be the autosomes. So they are the non-sex chromosomes. And then the sex chromosomes would be chromosome pair number 23 in humans. And when we look at the chromosomal pair of chromosomes then, we look at two different types of chromosomes. There's X and Y chromosomes. The XX combination makes a female, and the XY combination determines a male. And what's important to understand about those, however, is that the XX combination is a true homologous pair. So the genes that we find on X, we do not necessarily find on Y. So the XX combination is a homologous combination. But the XY combination is not truly homologous because the X has its own chromosome, or excuse me, own genes, and we do not have comparable genes on the Y chromosome. So that's not, even though we refer to it as a homologous pair, the XY combination is not truly homologous. And that will have certain effects. So when we look at the X and Y chromosomes then, the genes that we find on the X chromosome we refer to be as X-linked genes. And those that we find on the Y chromosome are Y-linked genes. So there are relatively few genes on the Y chromosomes, and those are largely related to what the sex of an individual will be. But I should let you know that the genes on the X chromosome, most of them have nothing to do with sexuality. Okay? But again, the point is that genes on the X are not found on the Y, and genes on the Y are not found on the X. So we are going to be interested in X-linked genes in our exercises as I proceed today. So let's go ahead and take an example. Let's say that there's an allele for blood clotting. And so the dominant allele here would be normal. The recessive allele, let's say, codes for hemophilia. And so as we look at the gene pairs of a female then, a female can be homozygous dominant, and that would be a normal female. A female could be heterozygous. And so she would still be normal because we're looking at typical dominance here. So it'd be just like looking at a heterozygote in any other situation. And in the sex chromosomes, we refer to a heterozygous female as a carrier. And then the only way a female can show the recessive phenotype is just like any other inheritance. She would have to be homozygous recessive for the recessive allele. So she would have to inherit two recessive alleles to show the recessive phenotype, just as in any other kind of inheritance. Now, when we compare that to males, we're looking at an XY combination. So again, there is a gene for blood clotting on the X chromosome, but that gene is not found on the Y chromosome. So in the XY combination, the X chromosome will carry the gene, the Y will not. So XY with the dominant allele on the X would be a normal male. And if the X has the recessive allele, then that would be an abnormal male, in this case having hemophilia. But what's important to understand about the male inheritance here is this, that X-linked traits come from the mother for one, but to show the recessive phenotype, the male only has to inherit one recessive allele, whereas females have to inherit two recessive alleles. Males only have to inherit one recessive allele. So the result of that is that males tend to show X-linked recessive phenotypes much more frequently than females do, because females have to inherit two recessive alleles, males only have to get one, and if they get one recessive allele, they show the recessive phenotype. So as long as you know something is X-linked, it actually makes pedigrees and so on pretty easy to do. So we, again, approach this just like we would any other genetics problems, but we would be focusing on the sex chromosomes in particular. So let's go ahead and look at a problem, and another gene that we find on the X chromosome, another X-linked trait, would be color blindness, various forms of color blindness. So again, a capital N here will represent normal vision, a lowercase will represent color blindness. So let's say that we have a normal male, and he marries and has children with a female that's a carrier for color blindness. So she would have normal vision, but she's hiding that colorblind allele. So what you do in these situations, and again, what would be the possible outcomes here? What would you expect to see in the children? Could any children possibly be colorblind? So again, what you would do is just set up a Punnett square, as we've done before. We would have our X and Y on one axis, and then our X's from the female on the other axis. So again, gametes would be carrying the X or a Y, or X's with the dominant allele or X's with the recessive allele. And then we would go ahead and combine as we would in any kind of Punnett square problem, see what our possible outcomes are here. And what we would have then is we would expect 1 out of 4 then to be colorblind. We would expect none of the girls, females, to be colorblind, although half we would expect to be carriers, and we would expect half of the males to be colorblind. So when you do these X-linked problems, you do want to be a little careful as to what's being asked in terms of how you answer it. In other words, if it was asking what fraction of the children you would expect to be colorblind, it would be 1 out of 4. If they were asking what fraction of the females would be colorblind, well, none. And if what fraction of the males would be colorblind, well, you'd say half, given this cross. So again, it's just like any other cross. You'd set it up like any other. Now, let's look at another example. Muscular dystrophy is another recessive trait which is carried on the X chromosome. So one of the questions here could be, well, is it possible for a normal man and a normal woman, is it possible that they could have a child with muscular dystrophy? So we would have to consider the different genotypes here and what the possible outcomes could be. So as we look at this problem then, a normal man would have to be an X with a dominant allele, because if he had the recessive allele, he would have muscular dystrophy. He wouldn't be normal. You would see that. And with a woman, she could be homozygous for the normal gene, but she could also be a carrier. She could be heterozygous, and she could be hiding that allele. So again, to answer this question, we would actually have to look at 2 different crosses. We would have to actually look at a cross between the normal male and the homozygous dominant female. And in that case, it would not be possible to have a child with muscular dystrophy. We would expect none of the women and none of the men to end up with muscular dystrophy. But if we looked at a carrier female, heterozygous female then, and go ahead and filled out our Punnett square, again, we would see that it would be possible for half of the males to have muscular dystrophy. So we'd expect 1 out of 4 children to have muscular dystrophy, but half of the males to have muscular dystrophy. And again, half of the females then would be carriers of muscular dystrophy as well. So again, when it comes to males, the X chromosome has to come from mom, because it's the Y that determines maleness. So we basically men pass on the maleness to their male children, and the X-linked traits are going to come from mom. Okay, now let's go ahead and look at a pedigree. Let's look at a larger pedigree, this again for hemophilia. And so as we look at pedigrees again, the shaded traits will, in this case, are telling us who has hemophilia. And you may recall that hemophilia would be recessive to normal blood clotting. And we always want to, again, start with what we know, who shows the recessive phenotype. And one of the things here, we could look at this pedigree and we would know that the shaded trait is recessive because we don't see parents with hemophilia and we do see offspring with hemophilia. So remember that only recessive traits can be hidden. Now the other question here as you look at this pedigree might be, how do you know whether it's X-linked or not? Could it be autosomal? Could this be an autosomal recessive? Well, it potentially could be, but what you would want to do then is look at who's showing the recessive trait. In this case, this pedigree, only males are showing the recessive phenotype. And if it was fairly balanced between males and females, then you would expect this to be an autosomal recessive characteristic. But since only males are showing this, that suggests that it's an X-linked recessive trait. And that happens to be the case, of course, with hemophilia. So again, we always want to start with those that show the recessive phenotype. So what we would do here then is go ahead, and we know that they're males because they're squares. And if they show the recessive phenotype, they have to be carrying the recessive allele. So we could go ahead and put in our males that are hemophiliacs here. But we also know the normal males. The normal males, again, only have one X. They would have X's with the dominant allele there. So we can go ahead and put those in as well. So again, in this case, it actually is easier to do a problem that's X-linked, especially when it comes to males, because the male shows what allele they're carrying. If it's a normal male, he has a normal allele. If he shows hemophilia in this case, it's because he has the hemophiliac allele on the X chromosome. So then it would just be a problem of following X's and Y's in the appropriate direction, at least in terms of females following our X's to try to figure out what their genotypes are. So again, we have 2 males here that are hemophiliacs, so she had to have given them an X with that recessive allele, so she has to be heterozygous because she's normal. So again, she would be a carrier. And again, here we have 2 sons that are hemophiliacs, had to get that X from mom, so she's a carrier there. And again, we see a son here that's a hemophiliac, so that suggests that she's also a carrier. And then the only one we really, or two that we can't be positive of is this female here because she doesn't have any sons that are showing it, and down here. So we can't be positive of whether she's homozygous dominant or not. Okay, so again, in a lot of ways, X-linked problems are more convenient to work with. Now, there's another peculiarity related to sex chromosomes that has to do with what's known as lionization. As it turns out, in mammals, the XX condition is a lethal condition. And having two active X's is not a good thing. It's not a healthy thing. Now, if we're a male, that's no problem. We only have one X. But if you're a female, that's a potential problem because you don't want two functioning X's in your cells. You only want one functioning X. So what happens early in our embryonic or early in the embryonic development of females at the 2 or 4 cell stage, one X or the other is inactivated. So again, the XX condition is lethal in mammals. So we're talking about mammals here. This is not necessarily all animals, but in mammals. And so what happens in embryonic development, one X or the other will hypercoil. And remember, in hypercoiling of DNA, it becomes non-functional. So this non-functional X chromosome is referred to as a Barr body. You can actually see it in the nucleus. And as a result then, when we look at female mammals are a mosaic of X activity. So some parts of the body, one X is functioning, and another part of the body, another X is functioning. And again, this process of inactivating one X or the other is known as lionization. So one of the places where we can physically see this effect, this mosaic effect of X activity is in cats. And in cats, an X-linked trait for fur color is found where there are two dominant alleles. So the B allele codes for black fur, the Y allele codes for yellow or orange fur. And again, this can be a little confusing because this Y allele is an allele on the X chromosome. So as we look at genotypes here, if we look at males, males can either be black males or yellow males, depending on whether they're carrying the black or the yellow allele. Whereas females, if they have two black alleles, that would be a black female. If they have two of the Y alleles, that would produce yellow fur, so it would be a yellow female. But if they were heterozygous for the two, again, if you combine these, what we will end up with is what's known as a calico cat or a tortoiseshell cat. Now, this doesn't completely explain the situation though, because if you were making black pigment and yellow pigment and you mix those together, you would expect that to be kind of a muddy brown color. So that doesn't really explain the situation very well. So as it turns out, this X activity is what accounts for the calico situation in a female. I should also tell you that there are occasionally calico males, but that's only because of an aberration. A calico male would be an XXY male instead of an XY male. And in humans, this is known as Klinefelter syndrome. And anyway, these would have reduced fertility. These would basically be sterile cats. So anyway, it wouldn't be a good breeder, let's put it that way. Okay, so let's go ahead and look then at how this heterozygous combination can result in a calico female. So again, we would start off with one cell. Basically, you'd be a zygote, a fertilized egg, and you'd be XX female here, heterozygous, or at least the cat would be. And the cells would divide. And let's say we get to the 4-cell stage. At this point then, lionization occurs. Randomly, one X or the other will be inactivated. And that means that the other X will be a functioning X. So again, either black or yellow fur then will function and be produced eventually when this gets to the skin. Now, once an X is inactivated, all progeny cells generated through mitosis will have the same X inactivated. So as these cells divide and undergo mitosis then, again, it will produce cells that would yield either yellow or orange fur or black fur. And eventually they distribute themselves throughout the animal, and some areas then will have yellow cells, fur-producing cells, others will have black-producing regions, and this is what eventually produces the mosaic or calico pattern that we see on the cat. So it's a combination then of being heterozygous and also activation or inactivation of one X or the other lionization. Okay, now I'm going to skip ahead here a little bit, and I want to talk about some other factors that can potentially affect phenotypic ratios. And so one of the things that can affect phenotypic ratios is what's known as polygenic inheritance. So again, normally what we're talking about, if we're talking about one gene with complete inheritance, we should see two phenotypes, dominant phenotype or recessive phenotype. But polygenic inheritance would be traits that are affected by multiple genes. And when there are multiple genes involved, we don't see two distinct phenotypes, we'll see a gradation of phenotypes from one extreme to another. So an example of this would be human height. Human height is affected by a number of environmental factors as well as a number of different genes. So you have people that are extremely short, extremely tall, and a wide range of heights in between that. So that's typical of polygenic inheritance. Incomplete dominance is where the heterozygous shows an intermediate form. We talked about this with flowers, where you could have red flowers, pink flowers, and white flowers. So typically, if you're talking about a single gene with incomplete dominance, that would be 3 phenotypes. We've also talked about examples where instead of 2 alleles, a dominant recessive allele, you could have multiple alleles. So you may remember blood typing there. In that situation, again, we had, instead of just 2 or 3 phenotypes, we had a number of phenotypes. So the more alleles are involved, the more phenotypes you can get. What's known as conditional gene expression can also affect the phenotypes that you see. For example, in Siamese cats, in their fur color, there's a gene that's affected by temperature. So that gene will be producing a brown pigment at a higher temperature, and the protein changes shape at lower temperatures and looks black. So Siamese cats are usually black at the tip of their nose, tip of the ears, and tip of their legs because they're cooler there, and so that protein then appears black in that region. Pleiotropy is where one gene affects a number of traits. So example, if you inherit a defective cartilage gene, it could affect your joints, it could affect your height, it could affect your skull shape, all kinds of different things, heart, muscle function, all kinds of things. Sex-limited traits are those that we tend to find only in one sex. So for example, if I inherited a predisposition to ovarian cancer, that couldn't affect me. I can't get ovarian cancer because I don't have ovaries, but it would affect a female. And then sex-influenced traits are those that are more likely to occur in one sex or another. So for example, male pattern baldness is affected by testosterone levels to some degree. So men are more likely to show that trait than women. So again, there are a number of different factors then that can affect the number of phenotypes that we see, although the basic inheritance is always the same. So that'll do it. See you till the next time.